Working at my family's pet store is always an adventure. Apart from cleaning out the cages, it's a pretty cool job, especially when any of the animals have babies. Like the other day, when 3 new hamsters were born.
As a kid who's just wild for animals, my friend James knows that most living things look pretty much like their parents.
Kids certainly resemble their parents. For starters, they usually have 2 arms and legs, 10 fingers and toes. But that's just the beginning. Your parents influence everything about you, from the shape of your nose to the texture of your hair. Want to know the long and short of why you look the way you do? Then let's get moving, 'cause this is the human body in action.
It's a little early to tell exactly what these baby hamsters will look like when they're grown. They could look like the mother, or maybe they'll look more like the father.
Whether you're talking hamsters or people, or even plants, the babies are called offspring. Still, no offspring is an exact copy of just one parent. Everybody gets a mix of characteristics from both of their parents. That's why you can end up with your mom's mouth and your dad's eyes.
Things that define who you are as an individual are known as traits. And traits that are passed down from parents to their children are called inherited traits.
Like the shape of your ears, how tall you are, or the color of your eyes. Where do these traits come from? They are inherited, passed down from generation to generation.
And it all starts with the microscopic building blocks of life, cells. Your body is made up of trillions of cells. The cell's control center is the nucleus. And inside every nucleus are tiny instruction manuals called genes. Genes don't just float around. They're carried on structures called chromosomes. Normal human body cells each contain 46 chromosomes. Each human chromosome can hold about 1,000 different genes. Genes are what pass inherited traits from parents to their children.
That's why the study of how certain traits pass from one generation to another is known as genetics.
This whole process is called heredity, and learning about it helps us know more about ourselves.
Hold on to your history books. Here comes a body blast from the past. For thousands of years, people have been breeding plants and animals to grow bigger or better, faster or stronger. But it wasn't until the 1860s that someone finally began to unravel the secret of how heredity really works. He was an Austrian monk named Gregor Mendel, and his amazing experiments with pea plants eventually changed the way people looked at themselves.
Mendel wondered how different plant traits, like stem height and seed shape and color, pass through generations. Altogether, the traits that you can see in an organism are called the phenotype. In people, traits like hair and eye color are part of a person's phenotype. And even though people can have many of the same traits, everyone's phenotype is unique. No two have all the same traits.
In one of his experiments, Mendel bred 2 tall pea plants, producing all tall offspring. And he bred 2 short pea plants, resulting in all short offspring.
Right, but when Mendel bred one of those new tall plants with one of the new short ones, guess what happened?
He got seeds that made medium-sized plants?
That's exactly what people thought back in Mendel's day. For example, it was thought that a tall parent and a short parent would make medium-sized kids. Seems logical, but no. In Mendel's experiment, the next generation of plants were all tall.
That's surprising.
Yep, and here's the most surprising thing. When those new tall plants were crossed, that's science speak for mated, and produced offspring, The next generation was a mixture of tall and short plants.
How'd that happen?
That's precisely what puzzled Mendel. Eventually, he came up with the theories which, to this day, are the basic principles of genetics. Mendel's first theory was that genes are inherited in pairs, one from each parent.
So one parent plant might contribute a tall gene, and the other plant a short gene?
Right, and remember that there are genes for all inherited traits, not just tall and short. All those genes together make up the genotype of an organism. Humans have about 30,000 gene pairs in their genotypes.
Whoa! Genotype? Phenotype? How can you keep them straight?
Just remember that genes make up the genotype, the total genetic makeup of an organism. But you can't see a genotype. It's just a set of instructions. Traits you can see or observe in some way make up the phenotype. Simple. Now, when Mendel bred a tall plant with a short plant, the offspring were all tall. But he knew that they had one gene from each parent plant, tall and short. This led to Mendel's second important theory, that the tall gene inherited from one parent was stronger than the short gene inherited from the other. Today, scientists call the stronger gene the dominant gene. The other half of the gene pair is known as the recessive gene. And since these genes produce traits, that means certain traits are dominant while others are recessive.
If both genes for a trait are the same, like two dominant tall genes or two recessive short genes, the offspring is called purebred for that trait. But if the genes are different, like a plant that has one dominant gene, tall, and one recessive gene, short, then the offspring is a hybrid, which means it has a combination of both genes. Recessive traits can still be passed on to the next generation. Let's look at hamsters. Fur color is a trait. It's part of the phenotype. The instructions in the cells to make the fur color are part of the genotype. In hamsters, dark fur coloring is recessive and light coloring is dominant.
These are my two favorite hamsters, Pepper with dark fur and Sandy with light-colored fur. If they had babies, I wonder, what color would they be?
It depends on what genes Pepper and Sandy got from their parents. Pepper is purebred for color. That means she has two genes for dark fur. Sandy is also purebred. He has two genes for light-colored fur. Let's make this easier by drawing a Punnett square.
A what square?
A Punnett square. It's named after Reginald Punnett, an English scientist who studied the theories of Mendel. In the early part of the 20th century, Punnett helped prove that Mendel's theories were correct. A Punnett square is a diagram that shows the possible combinations of genes for a particular trait, resulting from a cross between two parents.
Like James's hamsters?
Exactly. In a Punnett square, the dominant trait is always a capital letter, and the recessive trait is always a small letter. We'll use the letter F since fur color is the trait we're looking at. The left side of our Punnett square will represent the color genes of Pepper, our dark-furred female hamster. We'll put two small f's there, since dark fur is recessive, and we know that Pepper has two genes for dark fur. The top of the square represents the fur color genes of Sandy, our male hamster. Two capital F's there, since light fur is a dominant trait. Now, it's just a matter of filling in the boxes. Each box gets two letters. That's one gene from each parent. It's easy to see that every one of these babies would be a hybrid, with one gene for dark fur and one gene for light fur. But remember, only the dominant trait is expressed. That means Pepper and Sandy can only have offspring with light fur. But let's say we waited for this new generation of hamsters to grow up and breed with one another. That means a capital F and a small f on both sides of the square.
Check it out! If two of those hybrid hamsters become parents, it really could make a fur ball of a different color. This time, even though both parents have light fur, each baby has a 1 in 4 chance of inheriting dark fur.
Though the gene scene may seem like a game of chance, there is some order here. When we talk about genes and chromosomes, it's important to keep in mind that people are not pea plants or hamsters. Most human traits are not as simple as those of hamster's fur color or the plants Mendel studied. Our genes don't necessarily add up to an either-or situation, light or dark, tall or short. Instead, genes interact in complex ways.
Tens of thousands of genes control all of the different traits that make us human. That leaves a lot of room for possibilities.
Genes are what make those possibilities real. The tiny instruction manuals embedded in each of your cells.
And what are they instructing the cells to do?
Make proteins, the building blocks of new cells. They also help trigger the chemical reactions that physically create a person's traits.
So, genes carry the formula for producing proteins, but it's the proteins that actually make your hair black or your eyes blue.
You got it. But to read the instruction manual, you have to crack the secret code.
The secret code?
Friends, may I introduce you to the very stuff that genes and chromosomes are made of? The keeper of the code, the one, the only, deoxyribonucleic acid.
Or DNA for short. DNA is a huge double-stranded molecule that looks like a twisted ladder.
DNA is what genes and chromosomes are made of. Different sections of DNA make up different genes. And all those genes together make up chromosomes.
When it comes to chromosomes, more doesn't mean better. The number of chromosomes an organism has doesn't determine how smart or big or complex it is. For instance, a cat has 19 pairs of chromosomes, and a dog has 39. So you might think, oh, well, that's because dogs are bigger than cats. But then how would you explain the fact that horses and hamsters each have 32 pairs of chromosomes? And Goldie here has 47 chromosome pairs, more than any of those other animals. It even has more than we do.
That's right. Humans have just 23 pairs of chromosomes, 46 chromosomes in all.
If human cells each have 46 chromosomes, then why doesn't a new baby end up with twice as many? 46 from each parent, right?
The body takes care of that with special cells called gametes, which contain only 23 chromosomes. Gametes are produced through a process called meiosis, where 4 new cells are created from one cell. Think of it this way. Most human cells have 46 chromosomes. But to make it easy, we'll start with just 4. During meiosis, the DNA in that cell duplicates, doubling the size of the chromosomes. Then, chromosomes that are alike line up in matching pairs. Next, the chromosomes split apart and the cell divides, forming 2 new cells. Finally, the chromosomes in those 2 cells split apart again and the cells divide again. The result is 4 new cells, gametes. Every gamete, called an egg in a female, and a sperm in a male, contains half the number of genes carried by every other cell in the body.
It's during meiosis that the genes for each trait are selected from the parents.
Since the actual traits we inherit depend on our specific DNA code, we paid a visit to science star Dr. Richard Fishel. With high technology and cutting-edge research, Dr. Fishel has taken the field of genetics in a direction Gregor Mendel never even dreamed of.
I'm Richard Fishel, and I'm a genetics researcher. DNA is deoxyribonucleic acid. DNA is the carrier of our hereditary information. We all look like our parents in some way. We have similar eye color, we have similar hair color, we have similar noses. That is all coded for in the DNA. When you look at the genes that all of us humans on the earth have, we are 99.99% identical. We just have some very, very small changes that make us look a little bit different and maybe even act a little bit different. The code of DNA is made up of a very specific alphabet, A, T, G, and C. A stands for adenine, T stands for thymine, G stands for guanine, and C stands for cytosine. Those alphabet codes then make up the string of DNA, and how they are arranged in that string of DNA makes up the book of life. In threes, you can determine what protein is going to come out of that. Now, that's where things get fun. To get from the code in the DNA to something that makes you have to have messengers. Once you have encoded your code on the messenger, then you can translate that into what really is important for the cell, and that's the protein. An amino acid is a building block of the proteins, and the proteins are made up of amino acids. Those 20 different amino acids have a little bit different properties, and they help the protein function differently. Understanding genes and their messengers and their proteins is what constitutes biology. Understanding biology will help us to understand when it goes wrong. When it goes wrong, we want to fix it.
Let's get up close and personal with DNA, the genetic superstar, in a busybody experiment.
For this experiment, you'll need a source of DNA, like dried peas. They'll do nicely.
Yeah, Gregor Mendel would probably approve of the peas too. You'll also need several graduated cylinders, some water, a strainer, several 250 milliliter beakers, a 50 milliliter beaker, rubbing alcohol, some table salt, meat tenderizer, dishwashing liquid, and a couple of spoons for stirring.
The dishwashing liquid and meat tenderizer will help us extract the DNA from our split peas.
And you should also have a notebook and pencil handy to record your observations. Finally, you'll need an adult to supervise you because you'll also need to use a blender. Okay, let's get started.
Within a cell, DNA is protected by membranes. Those membranes are made up of fat, among other things. To release the DNA, we must break up the fat in those membranes. DNA is also shielded by a coating of proteins inside the cell's nucleus.
Your mission is to break down the fats and proteins and get to the DNA that's inside the cells of your peas.
First, you'll need to create two solutions. To make the first solution, take the beaker labeled 1 and put your salt in it. Then measure out 90 milliliters of distilled water and add it to your salt. Stir it up until the salt dissolves. Now add 10 milliliters of dishwashing liquid and stir it gently until it's mixed well, and set the whole solution aside.
Now we need to make a solution that will help us cut through the proteins.
Right, and to do that, we'll use the meat tenderizer, which contains enzymes that can break down proteins. So, place 5 grams of the meat tenderizer and 95 milliliters of distilled water in another beaker. This one labeled 2. Once again, mix them well and set the beaker aside. Now it's time for the peas. Go ahead and put your peas in the blender and add 100 milliliters of distilled water. Blend these on high for 30 seconds.
Sounds like we're making pea soup.
Well, you could say that. Now pour all your pea soup into a beaker. Then measure 50 milliliters and pour that back into the rinsed out blender. Now add 100 milliliters of the detergent and salt solution from beaker number 1. Carefully blend your mixture for about 30 seconds on a high setting.
Now pour the resulting pea soup through a strainer into a clean 250 milliliter beaker.
Now it's time for our protein-busting solution. Pour about 25 milliliters of the solution in beaker number 2 into the pea soup solution.
Now you need to stir very carefully for about 5 minutes. Be patient and don't stir too hard. If you stir too hard, you'll break up the DNA you're trying to find.
Now that all those pea cells are thoroughly broken down, we're ready to separate out the DNA. For this step, we'll use the rubbing alcohol. First, pour 20 milliliters of the pea soup solution into a 50 milliliter beaker. Now, Tilt the beaker and slowly pour 20 milliliters of the alcohol down the side of the jar so that it makes a layer on top of your soup. This needs to be done very carefully so you don't mix the two liquids.
See that white stringy stuff floating up to the top? Believe it or not, that is DNA, deoxyribonucleic acid, the genetic superstar.
Use a magnifying glass to take a closer look. It may be hard to believe, but you're looking at DNA, the genetic code for life. Within your DNA is all the information your body needs to live and reproduce. DNA molecules transmit that information by unzipping, actually splitting the twisted ladder in two, and then each strand makes an identical copy of itself. Unzipping and copying, unzipping and copying. DNA is the secret of how genes and chromosomes pass information from one cell to the next, and from generation to generation.
But what if a DNA molecule unzips and doesn't make an exact copy? What if that copy is slightly different? Then you get a mutation.
Chemical changes in the DNA can cause what is known as genetic mutation, and that can mean a change in traits. If that altered gene is passed to the next generation, it becomes part of the new organism's genotype and can show up in the organism's phenotype. While some human mutations cause hereditary diseases in people, others are actually beneficial, while most go completely unnoticed. Some variations help an organism to become more competitive in the struggle for survival. Without mutations in DNA, species couldn't evolve.
I wonder what kind of babies these guys might make.
Well, we know one thing for sure. They'll inherit their genes in pairs, one from each parent.
And we know that the traits that we can see are the phenotype. But the actual combination of all the genes is the genotype. Human genes are located on 23 pairs of structures called chromosomes within the nucleus of each and every one of our body cells. Like tiny instruction manuals, genes give cells the directions they need to make proteins.
The keeper of the code is DNA, what genes and chromosomes are made of. Unzipping and copying itself, it carries the blueprints for growth and reproduction.
How amazing babies are!
They are the wondrous product of genes and heredity, the pair that keeps the human body in action, generation after generation.